Okay, good evening, everybody. Can everybody hear me?
Yes.
Great. Thank you. All right, so for everybody who's not familiar with me, I might be a new face to some folks. I know we have a lot of folks dialing in. I'm Warren Anderson. I am part of the community. My son is Raceland Anderson. You guys might see some pictures of him later on. If you go onto our website, active2alliance.org, you'll definitely see some pictures. But I am the president of the nonprofit that we have, Active 2 Alliance, and we'd like to welcome you today to this presentation that we're going to present. Really, we're kind of emceeing all the information will be coming from a couple of really great experts that have been helping in the community. I don't intend to speak for very long. In fact, I'm going to go ahead and transition to introducing these two folks, since that's what you guys came here for. So I'll go ahead and start off with Dr. Musolino. Dr. Musolino, she's a pediatric neurologist over there at MGH. She's my family's favorite neurologist, so she works a lot with my son as well as a number of other individuals that are part of the community. She and Dr. Mark Lindsay, who is a cardiologist at MGH, they are leading some of the research into and providing insight into MSMDS. So that is the discussion topic for today. So we've asked them to speak on the state of the research. Back in May of last year, they presented a lot of information to individuals here in Boston regarding where they are in terms of their research and where we're going. We've asked them to speak today due to Rare Disease Day to provide an update to the community on the research that they are. So at this point, I'll go ahead and transition and send it over to Dr. Musolino.
Thank you so much, Warren, and thank you, the Active 2 Alliance, for giving us this invitation and such a special day. This is Rare Disease Day. It's been a day that has been mobilizing the world around rare diseases, which, as you guys know, affects a lot of people. I see in the people that are attending that we have our late night European families joining. So I guess this is around like 11 or almost midnight. So very nice to see you. And it's a privilege to be able to share and continue to share the journey and the research and the clinical and the care of these kids and adults affected by ACTA2 MSMDS. So we want to leave a lot of time for questions with Dr. Lindsay. So we prepare a kind of short presentation, and we're going to try to briefly walk you through what the efforts and research, not only on our group, but in much larger collaborations that have been initiated are at the moment, so you know a little bit of what's happening in our neck of the woods. This is a very large group. Each of these people have their own labs with their own scientists. Some of them, as you can see, like Mark, me, Raj, are also clinicians and that see patients. I'm the neurologist, Mark and Raj are the cardiologists, and you can see all the other experts in vascular genetic disorders, Carlos Ferreira, a geneticist in NIH, and then the scientists that really are cutting through the most advanced technologies, trying to help in this very rare disease, and ultra rare even, on getting a therapeutic option for these patients. So this is a very large team. I'm in debt to the trust these people have put on. They have done a lot of work out of their own efforts and funding. And then slowly we've been getting a lot of support from NIH and a lot of support from this entity called Angea Biotherapeutics. It's a very tiny, a small biotech that is basically a person affected by a genetic vasculopathy that is trying to help people with genetic vasculopathies get their therapies. So we're targeting the genes. That's why we call it a vascular gene therapy program, but we're doing it from the DNA level to all the way to the small molecule level. So it's vascular gene targeted therapies. And Mark is going to tell you a little bit of the update. We shared some of that in May, but we've done a lot of work since, and it will be great to share that with you. And we truly, truly, truly believe every patient, every journey counts, and we'll try to develop treatments for one patient at a time. And when we say with all in mind is because when we do get to treat patients with these severe conditions, we truly open up and enable the tools that we are using for many more other conditions that are less severe, where the risk is not justified for those therapies. So very brief introduction. You guys are the experts. I see who is in the call. But for those that are watching the recording, what is ACTA2 smooth muscle vasculopathy and what is MSMDS? It is caused by a change in one single letter of the DNA in the ACTA2 gene. It changes one amino acid, which is the arginine in that position, 179 of the gene ACTA2, which is acting for the smooth muscle cells. And that changing one copy of the 2 copies that everybody has is enough to alter the function of the smooth muscle cells. We will keep saying that this can be diagnosed at birth, and it's the testimony of the advocacy group, the Facebook group, the A2A, and YGSA that since the disease has been put in the map by Dr. Milowicz in 2010, and then the first natural history in 2018, the advocacy of the families, making everybody aware that a baby that has congenital mydriasis, so pupils that are not reactive to light, and the presence of a patent ductus arteriosus or an aorta-pulmonary window, which is a communication that is wider between the aorta and the pulmonary arteries, at birth, this is MSMDS, and it needs to get genetic testing. So we know that we can get to these patients very early on, and I think that in itself is changing dramatically what happens to these patients and to these kids, because there is a lot of measures in a standard of care that we can take to minimize the risk of stroke, for example. We know the strokes don't happen in the babies, except for the very deep white matter, which is around the time of birth, but the large strokes that are disabling don't happen until the kids at least start walking, and some of them go even much, much further out in life. And then the next big thread from the vasculopathy, the vascular disease, is the aortic dissections in the second decade of life. And almost as a sine qua non condition around 10 years of age, either the aortas reach the diameter where they need a replacement, or they have already started to have events of dissections that require surgical or medical treatment. We do know that a lot of what we are doing may not be able to prevent at the end the neurological consequences of a stroke. And many of you here also in the call have experienced the uncertainty and the disability of having to suffer recurrent type of strokes. And we are a referral center for the disease, and I think that has helped a lot develop understanding about the disease by caring for these patients during crisis and during the good times. And we also know that without a gene-targeted therapy, it's going to be very hard to undo the vascular effect of this pathogenic variant in the ACTA2 gene. Thanks to Diana and her team, and this is Dr. Regalado, genetic counselor in her team, that put together the first 33 patients in 2018 and kind of show that many of these patients were really dying early in life, that strokes were happening primarily in the first decade of life, and that the aortic dissections, as we were saying, are happening in the teen years. And this is aortic dissections and replacement, surgical replacement to prevent the aortic dissections. So the way that we are thinking about therapies for MSMDS, in particular ACTA2 arginine 179, which is the most severe form, is there is a period before the large strokes are happening, way before the aortas are most dilating, which we think is a preventive therapy treatment. So if we can fix the vessels in this early age, we probably can prevent a lot of the disease, both in the vasculature and the systemic, the parts of the body that, like the gut and the lungs and the bladder that also have a smooth muscle, as you guys know, are affected in the disease as well. Therefore, the name of the disease, multisystemic smooth muscle dysfunction syndrome. But we do believe that there is a lot of opportunity for rescue therapies, for halting that dilatation of the aorta, for remodeling these vessels in the brain, allowing new vessels to be formed, if we can make the smooth muscle cells be a little bit more normalized in function. We did work also putting together our cohorts with Diana's Milowick's cohort, and in 2021 in neurology, we found that by looking at all the MRIs from babies, children, adults with the particular variants in the arginine 179 causing MSMDS, we did find that that narrowing in the vessels in the brain is not severe at birth. This is at 10 days of life, and that it develops over time. And when you look at how it develops in most patients, it is happening early in childhood, that these vessels are narrowing in the brain. That never happens in children without this variant, without ACTA2 MSMDS. And the most important thing is that we know that before this gets really critically narrow, and that's kind of like more than 70% narrowing of that terminal ICA, there are no arterial ischemic strokes. So only the strokes we've been seeing are when this gets really critically narrowed. And as you may know, there are people still asking, do we need to do surgery in these patients, and try to do the revascularization surgeries? And we still don't have an answer. And in fact, it's part of the research that we're doing. We have worked with the YGSA group, MSMDS working group, and the families also that were working in the A2A Foundation to run a patient-reported outcome and a quality of life survey that we finished in 2023. This is being published now or shown in the Child Neurology Society last year in November, and the manuscript, thanks to Anna, is being prepared for publication. And what we found, and this I think is good news, guys, that in the Regalado paper, the first natural history study, patients were being, as a median, diagnosed around 11 years of age. And since then, only since 2018, it's only 5 years until we ran this survey, now most patients are being diagnosed before 3 years of age. And I want to cheer the community here, because this is thanks to you raising awareness, making people aware that this condition can be diagnosed, what are the features, and how to ask for genetic testing, and the work of a lot of physicians, though, of course, doing the publications that allow that to happen from the medical side. We also ask people to kind of categorize what is the disease burden and what is the most, the things that are impacting mostly the quality of life. And we found very much what we knew, that the neurological and the cardiovascular systems, followed by the lung, and in intolerance to exercise, are the most disabling ones, ranked by the patients or the parents of the patients. But there is also burden from other organs as well. And one thing that was very clear and is under yet study is that there is a significant impact. There are 37 patients, and of those, you can see up to 27 patients have reported developmental delays and difficulties with different areas of neurological function. So indicating that there is a lot that can be improved. And that's how we think when we are developing therapies. What matters to the patients the most? What will be something impactful and meaningful if we were to develop a therapy? What needs to change? What needs to be prevented? I mentioned this briefly. There is a surgery that is done in Moya Moya, which is not exactly correlate to the MSMDS vasculopathy we see in the brain. The main feature is we don't see a lot of new vessels in MSMDS ACTA2. And the vessels narrow, and there are parts of the brain that are losing blood flow here by this white area. And what the surgery does is bring this vessel from outside the skull and puts it in contact with the brain. And over a month, usually around 6 months, those vessels from outside the brain make new vessels to cover that patch of the brain that was having difficulties with blood flow. And the question is this helpful? Does it prevent strokes or brain injury or atrophy? And so far, we don't have the answer, and there is only less than 10 cases published in total. And the answer in the publications is 50-50. 50% of the cases have good outcome, 50% of the cases don't have a good outcome. So Angela Cass, who's also part of the board member of the A2A Foundation, has done on her own a whole mapping of which kids got the surgery, at what age, and what physicians intervene on that. And we're running in parallel a survey that is going to get to us, to you guys, the ones that have had the surgery, to report how things are for you from the perspective of the patient and how the clinicians rank the outcomes. And we will have all the images to also tell if the surgery is helping having the brain less injury, or if the brain is still, despite even new vessels, continues to suffer. And we're hoping to complete this research this year. And I'm going to pass it on to Mark, who has been the critical and the expert in ACTA2 vasculopathies in developing the next therapies for this disease. And I'll pass it on here so he can tell you more about it.
Thanks a lot. Actually, Patty's the expert. Yeah, so you know, we began this, Dr. Musolino and I began you know, working on this problem some time ago, and it started with the development of an animal model for the disorder. And so we've done a lot of characterization of this animal model, but basically it's a mouse that has an engineered gene change that's exactly like the children. And it changes that arginine 179 to a histidine. And so it really nicely, you know, acts in many ways like the human disorder. So we can see, for instance, narrowing of some of the arteries in the brain. We can see aortic dilation when we do mouse imaging. We can look at the size of their aortas over time. And they also have premature lethality. So we know that that's something that we think is happening in the mice because of the gut, and not necessarily from the vasculature, but it does allow us to test and see, you know, how a possible therapeutic could work. And so it's a really big step, and we're not the only one working on this. You know, Dr. Milowitz's lab in Texas also has an animal model that they've been making great progress on and have published. And so, yeah, this is a prerequisite for when you want to develop a therapeutic. So, so I think Dr. Musolino had this wonderful idea to say, can we really go after a gene therapy? This is you know, a condition that the gene change is often the same in many of the children, and therefore it may be you know, targetable by some of the new technology these days, the ability to actually fix a gene and a particular spelling error in the gene through the use of CRISPR enzyme, which basically is a platform for bringing in a protein into a very specific part of the genome. And then you can pick different types of enzymes that can modify DNA. And in this case, the kind of enzyme we want is called a adenine base editor, or an ABE. So just advance here. This requires a lot of work and a lot of people. And so our group, and through the resources that Patty described earlier, and through the support of our institutions, has been able to bring together a big team to work on ACTA2, and also, you know, other rare vasculopathies. But this right now is really a flagship program for us. So through the help of NGV innovation, and you know, and through our own work, we've been able to get a pretty big group of people working on this. So let me just take you through how these things are done. This is the initial work of the Kleinsteiber lab, who was Ben is a protein biochemist and expert at CRISPR engineering. So basically, first you need to model that gene change in cells. And so a cell line that had a specific gene change was developed so that that way we could target the mutation with different enzymes that could revert that particular mutation. It requires a lot of optimization because the enzymes are somewhat imprecise, and you want to be able to change the exact spelling error and not hit an adjacent adenine that causes what we call bystanders. And so through really a series of iteration using the cell lines, Ben's lab was able to create a really high degree of on-target editing without local off-target editing. And this is all modeled in an in vitro system, not yet in an animal. And so once this was developed, then we want to be able to put that in our animal model that I showed you before to see if we get an effectiveness. And really one of the challenges has always been a big challenge in gene therapy is delivering to the cells you want to hit. And in this case, the cells in the body are the smooth muscle cells. And so you need a platform for bringing these base editors into the right cells. And so we've used both a traditional adenoviral. This is a virus or a pseudovirus that can carry cargo but doesn't infect or adjacent cells like our typical kind of viruses we think of. But it can bring in an engineered product to a particular cell. And this particular one is called AAVPR, which was developed by Dr. Musolino and associates to really target the neurovascular specifically, but it also has a nice effect of hitting other smooth muscle cells. So really nicely, and this is data we shared in May, work in large part of Savydas, that it actually does both extend lifespan. So when given at either day 3 of life, which is sort of a imitation in mice of an infantile delivery, or day 14, which is more of a young child, young adolescent model, that it does in fact rescue multiple aspects of the condition. In this case, exercise tolerance, as shown by this thing we call the rotarod. It's just an apparatus that the mice run on to give us a sense of how well they exercise. And this is just the videos of a normal mouse that's in red, and then you can see how the ACTA2 mouse is just slower than his sibling. And then when treated with the ABE, they're really quite difficult to tell the difference between the two. So we have a nice exercise rescue, but importantly, advanced. We also get palliation of other phenotypes. So nice reduction in the aortic size, and both at the root and the ascending aorta. This is the part of the aorta right above the heart that we monitor really closely in clinic. And with this type of gene editing, we're able to see an improvement in those aortic diameters. And then also in the way the neurovasculature works. So we've modeled this functionally, but just looking here at just how the arteries look, the mutants are, have very thin-walled and lack the ability to contract, and as evidenced by them just kind of looking smooth. Whereas in controls and in the gene therapy treated mice, you really restore a normal look to the arterioles, where you have this wrinkled contractile appearance. And finally, we're starting to look at other phenotypes in the mouse. Ones we were delighted to see were also affected and improved by gene therapy. We know that the children have this congenital mydriasis, where you have dilated pupils at baseline, and this is risky to the retina over the long term because the eye can't protect the retina as well. And then on the left, these are actually pictures of mouse dilation, and you can see the mutant. Basically, you cannot see anything but the pupil which is different than the control where you see just a normal pupil. And in the gene therapy treated mice, we get nice restoration of pupillary contraction. So we think, this is a smooth muscle dependent process as well. And so it seems that at least in these animal models, we can restore some of that function as well. So just to kind of summarize the steps that are necessary to take this kind of work to patients. There's steps along the way. One is that you show an efficacy in vitro, and then you need to move on to an animal model that can model the physiology that we see in people. And we believe we have a good vector for delivery, that adenoviral vector. It's hitting smooth muscle cells, enough in the animal model to rescue the disorder, both in the things we want to help with, the aortic dilation, the neurovascular dysfunction, and prolonging lifespan. And then there'll be other steps in terms of looking at large animal models for toxicity and ex vivo studies to make sure that we can test that these are also going to be on target for humans. We're happy to say we had our initial interaction with the Food and Drug Administration in February, and was I think a really favorable meeting where we presented some of the approach and sort of asked questions about what hoops we would need to jump through to make sure that this was safe for the patients, and what their expectations are of that. So it's just a first step, but very, I think, important and helpful. So we're happy that that's moving forward.
So we have also been very privileged to be able to fund a true natural history study. So in those interactions with the FDA, one thing that became very clear is that to cross that finish line, to get a therapy to approval, there is a lot that needs to be known, since we are not going to have patients that don't get the drug, because it's an ultra rare disease. There are very few patients with this disease. The way that the regulatory agency, the FDA in this case here in the US, the EMA in Europe, look at the data is, can you do better than the disease does without any treatment? And that requires, and it's been asked by the FDA and recommended, to have a natural history study. So what is a natural history study? We told you about the Regalado first natural history is cross-sectional. You look one point in time, look back, see what you have, and try to tell what you know about the disease. We've done a little bit more in-depth retrospective characterization of the brain phenotype in the imaging paper that I was telling you, but there is a lot more to be done for the aorta, for the PDA, for the gut, for the bladder. So there is a need still for everybody to share their information so we can learn every aspect of the disease and what really is the burden of the disease. And then finally, because we are in 2024, and because now knowing a lot of the things that we just went through as the research advances, the clinical practice changes. They do not let you compare the effect of a treatment to a very old cohort where patients, as we were seeing, were diagnosed much older than the patients that we are talking about right now, that they are being treated right now as new patients appear and new babies get diagnosed. So there is a recommendation to have, in parallel to the development of that drug that we're trying to develop, to deliver in a clinical trial to the first patients in the first application, and that is called a prospective natural history study. And that means patients, while there is no treatment, are following a very systematic way, exactly the same way you will do if you're getting a treatment, but without an intervention. And in acquiring that data, it allows you to then really truly apples to apples and with the quality of acquisition where all the images, all the assessments happen by the same people, the same machines, you can really compare the data from one patient to another one. And not only that, which is more important in our disease here, is what happens in each patient individually over time. Because in ultra rare disease, each patient can become their own reference parameter. So if you have data for 2 years of how the disease is progressing and you can change the curve of that progression in the disease, that could be sufficient to say that the therapy is providing a benefit and hopefully get an approval of the therapy. So we were able to secure the funding to start the first natural history study. And as I was saying, it will have 2 parts. Everybody can participate in the retrospective natural history study. Everybody counts. And we need all the medical records that are available. And we have a very specific set of forms, plus the patient reported or parent reporter, caregiver reporter outcomes, which is how do you see the development and the quality of life and different aspects of the patient's life affected by the disease. So this part of the study, every person with a particular diagnosis of ACTA2 MSMDS can participate. In the prospective arm, we are starting with patients that have the arginine 179. In reality, we want to expand this prospective natural history study to other mutations and other variants in the ACTA2 in other genes. But at the moment, since the therapy we're trying to move forward is for particularly the most severe form, the 179 arginine, we are starting with those patients. And how does it work? And I'll show you a little bit of what the visits look like, but there is 2 parts. There is coming in person to visits for that perspective over a period of 3 years. There is only annual visits in person. And then you at home will have some wearables. So this is a watch that measures your activity in the heart, your activity in the muscles, and it can give a lot of information about what the body is doing. And those can be sent by mail and returned by mail and done at home. In combination with these remote visits, we will continue to acquire more continuous data about how the disease and what events have happened and how is it affecting the quality of life and the outcomes of these patients. So we chose a complex combinatory design because these kids are sick. So not everybody can travel. We want everybody to be able to participate in the retrospective. And for those coming, we wanted to decrease the burden of these visits to what is most common standard of care. And in fact, our IRB has approved the study, considering that the benefit to the participation in the study is that the studies that we are doing here at MGH could replace a standard of care. So meaning your doctors locally could use the studies that you are acquiring, that we are acquiring during the visit in that week. So here you have a schedule of what this study is going to look like, and we are ready to enroll. So I'll have a slide where you can contact someone, don't worry. But basically, we have to make sure that you have this thing called inclusion criteria. You can see that we want people with ACTA2 confirmed pathogenic variants. We do need at least one parent that can consent and read the study and understand what we are doing. That's a requirement of our IRB. And we also need the patients to be able to tolerate air travel or travel to the site. There are some patients that are local, of course, but for those that are not local, they need to be safe to get here. And basically, there is almost no exclusion criteria except pregnancy, which is something that the IRB requested, and having any medical condition that puts you at risk of travel when you have to come to Boston. And you can see there is an initial visit, a visit at 1 year, at 2 years, and at 3 years. These are the in-person in blue, and then in the green, you have the remote visits. This happen over Zoom, and we will have referrals for patients that want to participate, either the patients themselves, referring physicians, which have already put a lot of their patients in contact with us, and advocacy organizations. And on that, we're also teaming up with NORD, the National Rare Disease Association organization, to help us also with the reach that they have, get to a larger community of patients with ACTA2 and MSMDS than what the US Foundation has or the European groups have. So we're hoping that that way we also equitably offer opportunities to participate in the research to all patients that want to do so. And this is a very busy schedule for the families that are going to be participating in the prospective arm when you're coming to visits. And here's a description of what you're getting when you come. We have a multidisciplinary visit. It's all in the same place with all the doctors in the same room. And you guys being in Boston, some of you, and know how we do this. But the idea is that you see neurology, cardiology, hematology, a physiatrist, the rehab person, and a pulmonologist, together with our nurse practitioner, that will help us coordinate the care thereafter, all in the same visit. This is probably more a 2-hour visit. And then in the neurological testing, we're having brain imaging, and we have, our IRB has asked us specifically to modify and provide the protocols for sedation. In fact, we think more than half of the patients do not need sedation to get to MRIs, and we have Child Life that has helped us do this in our ACTA2 patients successfully. But for the little kids that may need sedation, we are doing a type of sedation that doesn't put the brain to sleep and is much safer in terms of the risk of stroke. And that's why we think this could also be of help, because you'll have an expert team getting the data that is sometimes necessary for clinical care. The MRIs happen only twice in the entire study. So it's one MRI in the first visit, 2 years later, there is another MRI. And we get the brain, the vessels in the head and the neck, and also the vessels in the chest, abdomen, and pelvis. That's the MRI of the aorta. We are also testing, giving what we learned from you guys with the survey, the neuropsychological evaluation, to see and really determine what is the impact in development and in neurological function of the disease itself, whether you have had or not a stroke or white matter injury. And of course, the exercise intolerance and the cardiovascular component, blood pressure, are getting evaluated with echoes. There is pulmonary function test, there is cardiorespiratory exercise. Each of these things is customized to the age group. So babies, of course, don't do certain things that the 4-year-olds do, that the 9-year-olds or the 15-year-olds can do. The study is aimed for a pediatric population to start. So the retrospective, everyone can participate, even patients that are not longer alive and adults. For the prospective, we are starting with babies to 18 years of age. And there is some blood work. We are doing all the invasive things during the sedation that happens during the MRIs, including an evaluation of the eye. And then we have the wearables that we were talking about that we are doing remotely, and all those visits in remote mode to decrease the burden of the families having to travel. This is what we call a sample schedule. So you arrive to Boston. If you're coming international, probably the day before. If you're local, maybe the same day, but we think ideally on Monday. And then there is a blood pressure monitor place that they leave for 24 hours. That goes in the arm of the patient. We get a very brief history here. This is primarily for the preparation for the sedation. You'll get the cardiovascular evaluations. If you haven't done remotely, you can also take some of the time to work with the CRCs and do the patient surveys during the time that you're in Boston. Wednesday is the imaging day. This is the day that half of the patients do it without sedation, the other halves do with sedation. And when you have sedation, we do the blood draw and a skin biopsy, it's a punch in the skin. And then for those of you that haven't done the skin biopsy, and then on Thursday is that multidisciplinary clinic. And then we have autonomic testing, the exercise testing. And then finally, on the last day, we do that neuropsychological evaluation. It's an abbreviated one. So we are hoping to do that in an hour. We put a little bit longer because some kids can do a lot more and want to, and we will test them with a larger battery. And then a physical therapist will help us estimate the motor function and the exercise function in more natural ways of these patients. And why is all of this necessary? Because if we have a therapy that is able to modify the exercise, we need to have quantifiable, reliable measures to show that the effect is robust and it applies to the disease. If you are interested, we are ready to start enrolling patients. We have funding for a limited capacity at the moment. We are putting together with NORD applications to other mechanisms, including NIH and the FDA, to get more funding, to get more patients involved in the natural history study. Diana is going to be the primary clinical coordinator, research coordinator for the study. Here's her email. She did this QR code, which apparently has all the information that you need. And you can contact her directly, and she will tell you a lot more about the study, and you can decide what kind of participation if you want to participate in. And I've said this before, we believe that in rare disease, we develop therapies for them, with them, and that's the reason why we're doing everything that we're doing. And I am thrilled and very happy to know that 3 of our known patients that have been in the ICU in the last couple of weeks are all doing much better, and they are all doing well. And that in itself is what keeps us going and doing the efforts that we're doing and trying to continue to move the research. And this is what Dr. Kinan, our pulmonologist, says: never ever give up on kids. I love it. It's simple, and it's what we are all doing, and you guys ahead of everybody. And this was a very memorable weekend for a lot of you, for us as well. And we are hoping that your example of how you're building this community can also expand to other rare genetic vasculopathies. A lot of those patients don't have a community and don't have a research team or several research teams like you guys have. So pass the torch, help other communities organize themselves, and bring how you're doing, clinicians, researchers, and families together, and you'll go much faster. With that, I think we take questions, if there is any in the chat, or you guys can also, I think, go on the microphone. Thank you.
Yeah, the protocol for this, if anybody has any questions, it's basically open mic. So just speak up if you have anything. We will call in.
I just have one question.
Sure.
Can you guys hear me? Sorry.
Yep, we can hear you.
If we were in Boston in May and we saw all the doctors, would that, like, if we signed up for this study, would that count as the initial visit, or would we have to do another one?
For this study, because everything gets standardized, including the imaging and stuff, they would not count. The studies would not count. What we propose is, if you have had your imaging recently, that you come in the study later on when the imaging is more meaningful to be acquired. So that's one possibility. Instead of starting right away, you can wait 6 months, a year, and start the study later, and have a more meaningful interval between the imaging that you are acquiring.
Okay. Yeah, he has not had recent brain imaging and things because it was pushed back for all sorts of reasons. So he has not had that. So I guess
yeah,
we wouldn't be able to enter later.
As you guys know very well, we've been very, very, very conservative with recommending doing imaging that requires sedation because of the risk that are associated with the sedation. So we have thought through what do we need to learn specifically for each patient, and there is flexibilities in the protocol when someone cannot tolerate an MRI or will not be safe to do an MRI, to acquire data at the same time but not have those endpoints. Ideally, for the comparison of how the disease is evolving in that kid and in comparison to the other children, you will need at least 2 time points with imaging. That's kind of like the minimum requirement to know if there is change or not in what's happening to your vasculature.
Good question,
Okay.
Dr. Musolino.
Oh, sorry.
Do you have anything else, Tia?
Yeah, I was just gonna say, but if our child has been sedated in the past for an MRI, but now we are thinking that maybe the use of child life could be more beneficial, would that be something that we could try for this, or would we have to do it the same way we've been doing it?
No, we are gonna try everybody on child life. We basically estimate that less than half of the patients, it will require that day. I don't know if you guys saw, they pretty much blocks the entire day, because a kid that is not doing it with sedation probably can do one part and then go have a coffee, eat lunch, come back and do the other part. So we are working with our team to get us flexibility on the scanners to throughout the week, try the way that we've done it when people visit also from abroad, try to complete the studies still without doing sedation. If for whatever reason with child life, with the goggles, with the parents in the room, we can complete the studies and we consider the risk of sedation reasonable. And again, the sedation is not the sedation that most commonly is done with MRI. We have modified so the brain is not fully asleep. So in theory, we kept the blood pressure control very strictly. We have not have at MGH so far, knock on wood, any events on any sedative anesthetics as of now. So we're hoping that the protocols that we currently use clinically are going to be safe enough also for the natural history study.
Okay, thank you.
Thank you.
One quick question. So your next engagement with the FDA, do you have a timeline that you're expecting for that milestone to occur, and what is the objective or objectives? What are the objectives of the next engagement?
That's a really good question. The next interaction is called pre-IND. So IND is the authorization to treat. So an interventional new drug, IND, that you can put in a human is the last step. To know exactly what the FDA wants to see in that IND package is a very large document where you show all the results and all the safety of the therapy in the small animals, in the large animals. We have this interaction called pre-IND. We did an interact where we asked them questions and they agree or tentatively agree to the proposed plan. We're executing that plan and our timeline is most likely in the fall this year. We will be doing a pre-IND. And in that one, we also gonna need the voice of the patient. That's how it's called, and the FDA requires it in rare diseases. So we're gonna be pitching to you guys to help us compile the burden of the disease and how the disease affects you in your own words. And there will be more communication through the foundation and the YGSA group on how you can participate on providing that voice for the pre-IND. And what does it take? There is a very large cost on securing manufacturing. So when you have to produce this for human quality, we've been doing everything for mouse quality, okay? So those are things that are expensive, but not so expensive as when you try to do something that is safe enough to put into a human. And that is called manufacturing. And in that manufacturing, what we need to secure is the funding to be able to do that manufacturing for the first few patients. And that is as part of what you put together when you go for this pre-IND. You tell the FDA, we're going to be doing this with these people, and here are all the processes that are going to take place. That's kind of like part of what is required. And that's why we're estimating that it's going to take us at least to the end of the summer or the early fall to be interacting again with the FDA.
Thank you. Anybody else have any questions?
No, I was just going to mention that I would be happy to talk to Tian about different ways she could prepare little Batman for MRI. Ella's been doing it since she's been 5 years old, the full scan each time.
Yeah. Yeah, and that's great. If you guys have resources, there's some resources the Child Life specialists have put together. There's also some YouTube videos. If you know those of you that have done this with Child Life, of how you can do it, it's absolutely recommended that you start doing it for not the study, for all reasons that we talk about, to be able to have your children not have to get sedation for the studies that are required to do clinical care. And it's a great offer.
Yeah, Penny, I'd love to get in touch with you. Levi just has a lot of medical trauma, as I'm sure a lot of our kiddos do. But Child Life has helped with some things when he had his surgery. And so I was like, maybe because there's no poking or prodding that comes along with MRIs, so it may be helpful. So Penny, I'll get with you another time to hear your suggestions and stuff.
Okay.
All right, the floor is back open. Anybody else have any questions before we adjourn for today?
I don't have a question. This is Dixie, by the way. I just wanted to say thank you to Dr. Lindsay and Dr. Musolino. It's always amazing to hear these updates, and it's really encouraging, and it brings us a lot of hope. So thank you for taking the time today, but more important, for taking the time over the last several years to help our families. We're really grateful.
It's a pleasure.
Pleasure, and thank you guys for the trust also in coming.
All right, last call. Anybody else have any questions? I'm sure they'll, if you can think of anything after this call, as usual, they have been really responsive to requests for information. If they have any questions, you guys already have their contact information. If you don't have it, please let myself know, or especially Dixie, since she's in regular communication with them. We can help spread their contact information.
There is a question in the chat. Do we know how much will be the manufacturing? And the answer is not fully confirmed, but we do have an estimate. And we are talking, prepare yourself, guys, at around $4 million to do the studies in the animals that require the safety and to be able to treat the first few patients. And that's the gene therapy. We did not talk about the small molecule work that we've been doing in parallel, which it's much more accessible, and it will be also available for patients that, for whatever reason, choice or not qualifying for the gene therapy because the disease is too advanced, or we are developing as well, and that is coming probably at the same time that this gene therapy is becoming available. So both, and it's important, is it the natural history study is developed in a way that you can come off the natural history study and go into the treatment arm whenever the therapy is approved to be treated in patients. So, and the idea is that natural history provides that trajectory of how the disease is evolving in your own child, and hopefully reference to what could be expected to be improved or changed or ameliorated in the disease by the therapy. So big steps, but a lot of hope, a lot of people with big hearts and a lot of trust on what we can do, helping us to get to that finish line. Yeah,
and that's the nice thing also is the natural history trial is agnostic or of the therapeutic. So, you know, the information that's gathered in that trial will be useful for the community, regardless of whether a gene therapy or, you know, works, because, you know, there are other therapeutics in the works. So that's the great thing about a natural history trial, is that that information is just super useful
for everybody. Yeah.
for whatever is being developed.
And it's is a great point that you bring, Mark. You will see in the consent, we are asking patients to give us permission to share the natural history data that is produced in this study with the advocacy groups. So our hope is that the advocacy groups themselves are the custodians of this database. So you can continue to work with whomever you want and have the possibility of share the data and save the work that is already been done, and also advance more rapidly towards whatever is the next objective or the next study. So after the study completes, the data will be transferred to the advocacy groups that request the data be shared with them. Patients are consenting to that. So that was part of our policy of how we want the data to be shared. So, and it's great to have it in the consent as well.
There is a question that's out there. I know that was a rough wag, the $4 million, but one, is that per person, and also
No, thanks goodness. No,
when are we shooting?
We don't know for how many, but we estimate that at the minimum, the first 3 patients will be treated, and all the work that has to be done until IND will be done with that fund. It doesn't have to be all before the summer. This is a process of 1.5 years, more or less. So, but yes, it's required and necessary. And we are working with Dr. Lindsay on a mechanism at NIH called Urgent. So you guys can take a look. And we're going to put this therapy, gene therapy, into that mechanism to see if we can get the support from NIH to do both the manufacturing and the clinical trial. And at the same time, we're fundraising with all other mechanisms that we have, philanthropy, et cetera, to be able to continue going at the speed that we're going. And if hopefully we get support from NIH, be able to do a full trial with many patients, et cetera, et cetera. So we are tapping on everything that we know exists. If you have any sources of funding or anything that you think we should be engaged with, please let us know. We are working really hard to really also put the research out there so people can contribute and collaborate.
I have a few more questions in the chat. One says, you mentioned more advanced disease. Is there an idea of what age will be able to benefit from the gene therapy, teenagers and adults too?
So we don't have that data yet, and that will depend on the FDA and how they see the data. So the FDA basically allows you or not to treat someone that has mild disease, severe disease, because they look at the whole package and they say the risk is not justified. Okay, so the equation of what patients can be treated, we elaborate what we believe. We have to show them in the mouse that we are able to modify the disease if we treat them, and we show you 2 time points, no? The babies, the baby pups of the mice, and they get the most benefit. And then we treat it like kind of juvenile mice, and they still get benefit. It's not as robust. And we are treating now older mice, and we don't know if we can help them yet until we do those experiments. So all of that data, plus everything in the safety side, how dangerous it is to do what we're doing, it gets kind of put on a scale, and the FDA decides what kind of patients can be justified to treat first. Because that's what they also ask you when you're doing something like this, is that you treat the first patients, you gather the data, you go back to them, you tell them it's safe or not, it's helping, not helping, and then they enable you to treat more patients. It's usually a staggered authorization, if you ask. So all of those details depends on the data and how the FDA looks at the data, and they determine what patients can be treated. Don't have an answer. And sometimes I want to say this, the FDA asks you to treat someone that is really sick. And the reason is there's a lot of risk. And maybe we have to treat them sickest first to say that it's justify the risk. Our main concern, as you guys know, always with any medication in MSMDS, ACTA2, is that we don't want kids or patients to have a crisis when they're getting a treatment. And that could be any medication. And for us, crisis means the blood pressure goes down. If the blood pressure goes down, we know we can have a stroke, a cardiac arrest. It has happened. We have lost patients with the disease to those events. So the FDA will look at, is it justifiable? Is it risky? Can we afford to do that? And that's what is going to cut that who can get treated first, who can get treated next. So it's a process. It's very transparent, and it's all based on data. So, and they come up with their justifications that they're very strong, and that's what their function is, guys, is safety, is to make sure that humans are not being treated inadequately or too risky.
Gotcha. Okay. Another question is, do we have access to those advocacy groups with our history?
What do you mean, the advocacy groups?
Yeah, this question was from Wendy. Can you expound upon that,
For
Wendy?
the for like the natural history, is there going to be like one group where we can access all of the information, or is there going to be several different groups, and do we have access to those?
So the natural history data will come out from the MGH. There's a database, it's called Neurobank. It's used by NIH also to do a lot of studies across the country. And the database, in that database, when the study finishes and the data is published and stuff, the advocacy groups that have the show interest on having that data will have a discussion with investigators. If we consider you serious, A2A, like you guys are the main recipients, if you ask me, but it could be the Writer Foundation, it could be people that really are working towards helping these families. That data will be available to those advocacy groups. NORD will have the data too. Is that answering the question, Wendy?
Yes, partially. And then the other part would be like if you had mentioned something about like if we wanted to share information or like our medical records with, say, some team members that we're not a part of, or that we regularly see, will we have access to all of that in the database to present to somebody else? Or no, it's just whatever we have in our own records?
I'm not following you, but so the retrospect. So every patient that participates we take all the data that you have prior to coming to the study. That's the retrospective side. So all your medical records, every surgery, everything that happened to that patient, and then we continue to collect prospectively with that patient, the ones that can come to the prospective study. And then all of that is packaged. There are different forms for collecting the retrospective and for collecting the prospective, but all of that data of how the disease affects and what parameters and the echoes and all this stuff, that becomes available. So you can participate on the retrospective arm and that data will become available. It's de-identified, so you cannot identify the patient on the data. So that's a way, and it's a whole like security and patient privacy regulations that we follow to do these studies. And when the data is shared, is data that is de-identified, doesn't have the patient identifiable information.
Got it. Thank you.
No, you're welcome.
Diane, you had a couple of questions. I think one of the answers that Dr. Musolino provided earlier might have answered your first question. Did you have anything else?
Yeah,
on that?
she answered my first question, but I did ask another question about the study, like the natural history study. And it may be a dumb question, to be honest, but I didn't know if they knew what the like cost or what would fall on the family to pay for to do the study.
Nothing. They don't let us do that, and I'm very happy about it. The study, so when we say we secure the funds pay the kid, 2 adults to come with them to Boston, stay in the week, a stipend to live in the city and eat and those kind of things for the time, and all the procedures of the study. It's all covered in the fund of the natural history study. So the families do not encounter medical bills or anything. Everything that the insurance decides not to cover because they don't consider a standard of care, the fund covers the cost. It's not going to the patients.
But you guys will charge our insurance to try to get coverage for some of it, correct?
Yeah, this is mandatory for something that is, so there are procedures that are standard of care, there are others that are not. The ones that are standard of care, you have to charge the insurance, and then the insurance declines or not, and then automatically charges the fund, the research fund, if it's declined for whatever reason.
Okay, I just
It's the
wanted
whole
to check.
process that I'm minimally
But
clear, but
The idea is that if you're going to your regular pediatric cardiologist and you're getting an echo anyway, we don't want you to have to get an
True.
extra echo with your child. We just want to do it and then provide, you'll go home with that information, and the insurance company would be paying for that either way. So they should. So that's
Okay.
that's the concept for things that
Okay,
are
so
appropriate for MSM.
okay, so if they've had some of these studies done, will they still have to have them redone in Boston? Because I know like Levi's had several echoes in the past few months because of his heart surgery that he had in August. And then he has one in April too. So I know that like he has them very frequently. So would that be something that you guys would just get from his doctor and not have to redo, or would we have to redo it at Boston?
so go ahead, Mark.
Yeah, it depends. So like if Levi was in the study and he was due for an echo and his pediatric cardiologist there and wanted an echo and you're already coming, then there's no reason to do it twice. So we would just say, why don't you do it with us? We're going to give them the results. But if they want one sooner,
They do it.
they want one sooner. Some pediatric cardiologists, like, they want it done with them in their clinic. And, those are the things that we can't really predict, but I think we would need to do the studies here for the kids that are in the trial. We would work very hard to make sure that it's not duplicated. But
Okay.
And that's the other reason we know echoes. We recommend echoes in most kids every 6 months when we start seeing the aortas. And the study only has an annual one. Yes, and then the imaging of the heart, and we are very familiar with the disease, and a lot of these children are having procedures for very necessary medical needs. And that means that some visits may not happen because they have to be in the hospital, we have to delay them. There is a lot of flexibility in the study because the reality is this is what the disease is. And at the same time, we designed the study to be able to provide the most comprehensive care by an expert team. So the local teams can benefit from whatever we can do and don't have to carry some of the risk of sedation or have, for example, the multidisciplinary team consultation available to have the assessments that we have for the kids. And the families do get the MRIs, the echoes, the notes, all of that goes into your medical record. So you will have it available. Everybody that can see the medical records and you authorize to see the medical records will have it available. It's not only research. We, in particular, strive to get the medical reading and all the reports in this study to be able to provide to the patients what they need for a standard of care.
Thank you.
You're welcome.
Okay, any other questions? Looks like the questions are slowing to a crawl on the chat. Okay, nothing heard. Thank you very much, Dr. Musolino and Dr. Lindsay. I echo Dixie's sentiments. It's always been a pleasure working with you two, and we appreciate your commitment to the community. Thank you very much for donating some of your time tonight to update the community. We really appreciate it.
thanks, everyone.
Thank you for having us.
Thank you.
Have a
Thank
good
you.
evening. Have a good morning for the Europeans.
Good night.
Bye.
Thank you.